Welcome student in the 7th week of our SWAM course Anatomy of Flowering Plants. Till now we have discussed about the primary structures, how were the different cellular structures and tissue formation they were organized in a plant body, and we have also discussed about the secondary structures, how cambium was giving rise to different secondary structures and how they were organized in a plant. Now we will in this module, we will discuss about the anomalous structure. Now, anomalous structure means those structures which are not normal, like whatever is there in primary organization and whatever comes out in the secondary growth, if it is not normal as all we have discussed, it will come under the category of anomalous structure. So, lot of anomaly is there in plant, different species of plant. So, that all we will try to analyze. Now, different anomalous structures can originate by different ways. One is that they could be either present from the initial development of the plant, even in the primary structures, those anomalous structures could be found, or many of them arise during secondary growth formation. Okay, when normal cambia give rise to secondary growth, that time also different anomalous secondary structures could arise and they will form the anomaly in the plant. So like, see, apart from that, there could be many things also, like if in dicot, normally we know that in dicot, the vascular bundles are arranged in the form of ring. Okay. Suppose this is the epidermis and then you have cortex. Okay. And then lot of vascular bundles are arranged in the form of ring. Similarly, in monocot also you will have a small hypodermis and then a complete ground tissue in which the vascular bundles will be scattered. In this you will have epidermis, cortex, hypodermis will be there, then endodermis, pericycle and pith will be there. So, all these things will be there, but in this monocot lot of vascular bundles will be scattered. Now, this is the normal structural organization of a dicot or a monocot plant, but suppose if that organization is disturbed and a different variation is found, that could be an anomaly, which is, see, it's not normally present in all the dicot plant or all the monocot plant, but in some species it has been reported that in dicot scattered vascular bundles are found. Okay, so suppose this type of arrangement you find in dicot, so it will be an anomalous structure. Similarly, if you find an organized or arranged vascular bundle in monocot, then that could be a anomalous structure. Okay, so these are reported in some families, like in dicot, in Thalictrum species, which is found in Ranunculales, this type of scattered vascular bundle is observed. Similarly, in Podophyllum of family Burbidaceae and Papaver of Papaveraceae family, these scattered vascular bundles are found. Similarly, in monocot, vascular bundles are arranged in the form of ring in Thamus communis of family Dioscuraceae. Okay. So, these are also anomalous structure which might have originated from their primary growth itself. Similarly, presence of cortical bundles and presence of medullary bundles are also one of the very important anomalous feature which is found in plants. See, cortical bundle, from the name itself you may make out that it is found in cortex, and similarly medullary bundles it is found in pith, because pith is also known as medulla. So, like in this you have normal, suppose you have normal arrangement of vascular bundle, and if you find some vascular bundle in the cortex region, that will be an anomalous feature. Now, generally these bundles are not the only bundles which are found in those plants. Those plants will definitely have their own normal vascular bundles, but along with that they have cortical bundles in the cortex region. Similarly, in the pith region, suppose this is the pith portion, okay. and in that you have vascular bundles. So that will be medullary bundles. This is the cortical bundles and this is the medullary bundles. So there are many plants in which lot of medullary bundles have been reported. In that also normal vascular bundles might be there. Okay, so they might be responsible for the normal secondary growth and whatever other processes are there. But along with that, they also have extra bundles. Similarly, inverted bundles could also be present. Now, inverted means in which the position of phloem is inverted as compared to xylem. See, normally xylem is found on the inner side and phloem is found on the outer side. But if that condition reverts and phloem is found on the lower side, so that condition is also not normal, so that is also found. So Reumex species of family Polygonaceae, this type of inverted vascular bundles are observed. Now we will discuss in somewhat detail some of the important anomalous structures which are there and what are the reason behind the formation of these anomalous structure. See, this is also important to note that in one plant you can have many different anomalous structures also. Okay, it might contain only one, like suppose it might contain only medullary bundles, but along with that it can have anomalous secondary growth. So different types of anomalous structures could be formed even in one plant. Okay, so let's try to discuss some of the important one. Now in this also we have discussed before that how these anomaly will arise. It can arise due to abnormality in the cambium development also, like cambium which is formed could be normal, but it is giving rise to abnormal secondary structures in terms of xylem and phloem. That means either it is forming xylem and phloem in wrong locations or in wrong orientation or in different proportion in which it should actually occur, or sometimes the cambium is itself is the normal cambium is not active and there arises an abnormal cambium, means a cambium which might arise at some other location, and it may also give rise to different proportions of secondary tissues, different orientation of these secondary tissues. Now first important plant which we will discuss is Nyctanthus. So in this there is formation of accessory vascular bundles. Okay, so first of all we will start our discussion with those vascular bundles which are present from the primary structures itself. Okay, we will discuss the abnormality of cambia in the next lecture. So in this one we will start with accessory vascular bundles. So Nyctanthus is one of the very important plant in which you will find accessory vascular bundle, and in this you will find the presence of cortical bundles. That means the vascular bundles which are found in the cortex. See, now Nyctanthus plant, I think so you all might have seen. It's beautiful. It contains, it's a fragrant, it produces fragrant flowers which has lot of smell, means scent is there, a very beautiful scent is there in these flowers, and you would have seen a white color flower with the yellow or orange mark at the center, and this we also say it as parijat. Okay, and when you will observe the stem of this flower, you can see here the stem is actually quadrangular. See, it is not circular. Okay, so it has margin like this. It doesn't have margin in the circular form. It has a square or rectangular margin, you can say. So it is quadrangular, and what at the four corners, what you will see when you will cut the transverse section of this stem. Okay, so this is basically TS of the stem of this Nyctanthus plant. You will see that at the corners, four cortical bundles will be there. Okay, but when we draw actually transverse section, we also find lot of other elements. See, you will have collenchyma, as we have studied before that in dicot, a hypodermis will be there, that will be collenchymatous. So in this case also you will have collenchyma. Okay, then you will have parenchyma also in the cortex. So hypodermis will be followed by cortex. And you will find hairs also, trichomes. Generally these trichomes are found in the dicot plant. Now at the four corners you will find these cortical bundles. Okay. In this cortical bundle you will find cambium and cambium, okay, and xylem and phloem. So this is the cortical bundle. Now what happens to the inner structures? See, normally it will also have its own vascular bundle. So those vascular bundles, as I have told you that in dicot plant, generally those vascular bundles will, as soon as the plant has attained a particular height and has attained a particular growth, it will start forming secondary tissues. So when you will cut the transverse section, generally you will not find a normal ring-like arrangement of vascular bundle. You will find a continuous ring of xylem and phloem, which means that secondary growth formation has occurred, means a ring of cambium has formed and it is continuously forming xylem on the inner side and phloem on the outer side. So here also generally the stems which we will take, definitely they will have given rise to secondary tissues. So here also you will find secondary phloem and secondary xylem. So normally when secondary growth will occur, the endodermis and pericycle, they all will be crushed. Even primary phloem will also be crushed. You will generally find a secondary phloem. Okay, so this is phloem. Basically it is secondary phloem. This is secondary xylem. This is primary xylem. Okay, here also like you can mark it as epidermis. Okay, then this one you can mark it as hypodermis, and it is made up of collenchyma. Then you have cortex, which will be made up of parenchyma. Okay, so this type of structure you will see. Now presence of cortical bundle is anomalous behavior, but inside cortical bundle also the organization which is there, that are also having lot of anomaly. So when you will draw this cortical bundle, suppose you make a bigger structure of this cortical bundle here. Let's change the color. So it will be okay. Few layers of cambium initials will be there, and then on the lower side you will have phloem. Okay, small cells. When you draw phloem, you should draw it the small cells to represent phloem, because as compared to xylem they are not bigger cells, and their walls are also not highly lignified. And when you will stain them, you can easily make out that I have told you that there are different stain. One stain is there which colors lignified structure, so you can easily see the xylem in those tissues. Okay, so this I am drawing xylem on the upper side and phloem is on the lower side. So this type of vascular bundle is known as inverted vascular bundle because it has xylem and phloem in different orientation. On the upper side also some sclerenchymatous tissues are there. However, many of the xylem tissues are also sclerenchymatous in origin. They have lignified cell wall. So you can see here, suppose this is the pith region, this is the lower side, and on this side you will have the epidermis. So what you can see, this is the phloem, okay, and this is the xylem. But in this case also you can see here that the lower one is metaxylem and the upper one is protoxylem. So this condition is exarch condition, okay. See, we generally when you will see the cellular diagram of these structures, you will see that these vessels will be marked as protoxylem or metaxylem because vessels are very easily, you can identify them in cellular structures because they are very big holes. So you can easily make out that if very big holes are there, it is metaxylem, and smaller holes will represent protoxylem, whereas other structures are also xylem, okay, and they will also have their protoxylem and metaxylem, but it is very easy to distinguish the protoxylem and metaxylem on the basis of vessels because they are very big structures. See, now this is also an anomaly because normally in stem you find endarch situation. See, in the primary structures also, these primary xylem, if you will observe this primary xylem, you will observe endarch, that means protoxylem towards the center and metaxylem on the peripheral side. But this is also an anomaly. Then inverted condition, inverted vascular bundle is also an anomalous behavior, that is phloem on the lower side and xylem on the upper side. So there are basically three anomalous structures or three anomalous behavior which you can see it is found in Nyctanthus. The one is the presence of cortical bundle itself, and in this bundle also the organization of these bundle is having anomaly as compared to the normal vascular bundles. It is having inverted orientation of xylem and phloem, and it is having exarch condition of xylem. So all these structures are present there. Now normally what might be the origin of these cortical bundles? It is said that they are generally leaf trace bundles. Now leaf trace also we have discussed before. Suppose if a leaf is coming out, okay, so it will also require vascular strands. Now the vascular strand which is entering inside the leaf is known as leaf trace. Now this, you can say that the normal strand which is coming out in the stem and it is moving in the, it is moving in the leaf is known as leaf trace, and the place where it will form, it will enter inside the leaf will form a gap, so it is leaf gap. So it is said that suppose this is the vascular bundle, this is a complete cylinder of vascular bundles, okay. Now suppose a small piece of this vascular bundle is entering the leaf, so in this way it will enter. Now what happens, this small vascular bundle instead of entering into the leaf when during leaf formation, okay, in the starting of the plant development itself, this doesn't enter into the leaf and it comes to lie in the cortical region of the stem, like it was coming out from here, but instead of going inside the leaf, it has lied inside the cortical region and has formed. And in this way, the orientation of xylem and phloem also gets inverted, and even the protoxylem and metaxylem, they are also inverted. So in this way, you can find this type of cortical bundle you can find in other families also, like in Crassiorhinaceae, Umbelliferae, Papilionaceae, Rutaceae. In few members, in some examples you can find them, but try to remember this Nyctanthus example. Now this is just the cellular diagram to tell you that how this whole structure will actually look like when you will observe it under microscope. See, this is the transverse section of a stem which I had prepared for your understanding. See, this way you will cut a very thin section of the stem, transverse section, okay, in this way, and then you will stain them with fluorogluconol. I have stained it, fluorogluconol, and HCl, okay, combination. Then you will see pink colored or red purple color tissues which are stained. So these tissues are dead tissues. If there is any sclerenchyma also, that will also be stained pink. So you can easily see a pith region in the center. Okay, a pith is there in the center. Then this is all secondary xylem. Primary xylem will be definitely below secondary xylem, but they are so less in numbers that, and you cannot even differentiate them also. Okay, and on the basis of size only you can differentiate. You can see here that vessels are there in this secondary structure. Now phloem is, definitely the phloem is not stained, okay, but you can easily make out that phloem will be on the above secondary xylem, the secondary phloem. So you can see a grey color patch of secondary phloem also. Primary phloem is almost crushed, and endodermis pericycle, they are also crushed. Sometimes pericycle patches could be there. If sclerenchyma is present in pericycle, those patches also you can see. Now this is a more magnified view. See, here also you can easily see those vessels. Now try to focus on the cortical bundle. See, here also you can see this is the xylem. Okay, small, very small vessels, okay, as compared to secondary tissue. That also I have told you that in primary tissue the vessels will be very small if you compare it with the secondary tissue. So if you compare it with the secondary xylem, the vessels which are present in the cortical bundle, because they are generally of primary nature, okay, because in that, although cambium is there, it will also give rise to some secondary tissues, but that is not that much as compared to the main cambium. So in this you can see very small vessels, and it is moved upward. The vessel size is keep on decreasing. So that will be protoxylem, and this will be a metaxylem. All rest of the, say here also you can see phloem. Okay. Now if we closely analyze, you will see that many other tissues. See, this is of parenchyma, cortex. Okay. Parenchyma, cortex is there. We can easily make out that some green color cells are there. So that might be chlorenchyma, because it's a green stem, so it might be performing. Here also you can see collenchyma. Okay. You can see that it is not green and it is compactly packed. So all these tissues you can see when you will cut the section. See, exactly you can see it here in the cellular diagram. You can see epidermis, then cuticle is definitely a thin layer, cuticle is there, trichomes are there, multicellular, then here xylem. Okay, so this will be protoxylem and this will be metaxylem. And then you will see, this is collenchyma, this is parenchyma, then secondary phloem is there, small crushed primary phloems could be there, crushed pericycle could be there, then cambium and this secondary xylem. Okay. Now next is the structure of Boravia, very important, in which you will find medullary bundles. Okay. So Boravia also, see in this multiple bundles you will find, if we try to see the structure of this Boravia, in this also definitely you will have epidermis and then collenchyma and then parenchyma. Okay. Then you will have endodermis and pericycle. Okay. So in this you will see two large bundles in the center. Okay, their phloem will be on the outer side and xylem will be on the inner side, which will be followed by 6 to 10 around, or 6 to 14 middle sized vascular bundles. Okay, in the middle region, and then smaller vascular bundles will be about 15 to 20 smaller vascular bundles will be on the outer side. So that means you will see vascular bundle in 3 rings. Okay, 2 bigger vascular bundles will be in the center, then followed by a center middle vascular bundles, then third ring will be of the smaller vascular bundle. Now the center vascular bundles are actually the medullary bundles. So This is anomalous behavior. Now during the time of secondary growth formation, the third ring, the outer ring of smaller vascular bundle behave as the normal vascular bundle, and they will form, their cambium will join and they will form secondary growth. However, in Boravia, you will also find another anomalous behavior, which is abnormality in secondary growth formation. So that we will study later when we will discuss about that topic. Now this is the cellular diagram, and this is the picture of the transverse section of a stem, which I have cut and I have stained them. You can see the presence of big medullary vascular bundles. You can see xylem on the inner side and phloem on the outer side. Here actually secondary growth formation have occurred on the outer side, that I have already told, that when a plant has grown up to a certain stage, then always you will see secondary growth formation. Then medullary bundles are also found in other plant species, like in Amaranthus. You will find lot of secondary bundles which will be present inside the pith region. Okay, lot of scattered. Here also you will see vascular bundles of different sizes scattered in the pith region. So this will all be xylem and this will be the phloem. So in this medullary bundle, see, in all the cases, these medullary bundles are normal in their orientation. They are conjoint, collateral and endarch condition. Okay, secondary growth formation doesn't happen. So generally, either if cambium is present, it will be very feebly developed, or almost it will be functionless or absent. So medullary bundles does not take part in secondary growth formation. See, all these features, even in Boravia also, you will find the normal conjoint, collateral and endarch. Now just try to recall what all these terms mean. Okay. Even in Mirabilis also, you can see lot of vascular bundles in the center. You can see some bigger vascular, generally some bigger vascular bundles are found in the center, and those which are on the periphery, they are smaller. And same conjoint, collateral and endarch condition. See, in the cellular diagram also you can see, you can see collenchymatous hypodermis, then chlorenchymatous parenchyma in the cortex, then star sheath is there, pericycle is there, okay, secondary phloem, secondary xylem, and these are the medullary, because the outermost vascular bundles will behave and give rise to secondary tissues. Similarly, in Bougainvillea also you will find lot of vascular bundles in the center. In this you will find some collenchymatous tissue on the boundaries, and then you will have cortex, and then in the center lot of vascular bundles will be there. So this is also a section of Bougainvillea in which you can see lot of vascular bundles, okay, which are scattered. This is also a transverse section in which you can see collenchyma patches are there, okay, and then chlorenchyma. See, it is in patches, it is not in continuous layer, and then you have chlorenchyma, you have endodermis, pericycle, then all these different sizes of vascular bundle in which you can easily see phloem is on the outer side, xylem on the inner side, and protoxylem is on the inner side, and metaxylem is on the outer side, that is the endarch condition. Similarly, in Achiranthus also, here you will see two vascular bundle in the center, two medullary vascular bundles, okay. Phloem is on the, in this case you will not find lot of vascular bundles, only two. So this is important that in Achiranthus you will find two medullary vascular bundles. So this is also having all the different layers. Outer vascular bundles, normal vascular bundles will be there, they will give rise to secondary tissues. In Cucurbita, okay, see Cucurbita also, see it has wavy margin because the stem has some ridges, okay. So in that ridges you will have collenchyma, okay, and then you will have endodermis and pericycle, all those layers will be there, okay. And in the center actually you will find vascular bundles in two rings. Okay, outer ring of smaller vascular bundles and bigger ring of, and make it bigger slightly. Now, one important feature which we have discussed before also, that the vascular bundle of cucurbita is collateral. Okay, that means phloem and cambium are present on both the sides. Okay, so here also you have cambium and phloem, and in this, and xylem is present in the center. So that means this is both cambium and this is phloem, and this is xylem. So you will find two rings of vascular bundles and collateral. Vascular bundles are collateral. You can see in the outer side you will have hypodermis, collenchymatous, then some cells will be parenchymatous, pericycle. Okay, pericycle will be made up of sclerenchyma, then endodermis, and see in this case you will see this is cambium on both the sides and phloem on either side and xylem in the center. Xylem will be endarched, see protoxylem on the lower side. So this is all about the accessory vascular bundles which are present. Now whatever structures I have discussed, in that either those structures are the only anomalous structures or they will have many multiple anomalous structures also, that we will discuss in the coming lectures. So for today, thank you.